So my name is Ty-Angelo Giorcelli, and this podcast is about how the coronavirus works on the cellular level and possible treatments that could reduce the severity of it. This podcast is a review of scientific articles, overview of the pathogenesis and treatment of SARS-CoV-2 for clinicians, a comprehensive literature review by Christian Voto, Paul Berkner, and Carol Brenner, all of which have PhDs in a medical practice, and SARS-CoV-2 treatment approaches, numerous options, no certainty for a versatile virus, by Simona Lacob and Diana Gabriella Lacob, both who work with institutes dealing either with medicine or infectious disease. 3 species of CoVs have been responsible for important and severe respiratory outbreaks in humans, namely the severe acute respiratory syndrome coronavirus, SARS-CoV, which led to the 2002-2003 outbreak, the Middle East respiratory syndrome coronavirus, MERS-CoV in 2012, and the severe acute respiratory syndrome coronavirus 2, SARS-CoV-2, associated with the ongoing pandemic also referred to as COVID-19. Coronaviruses are a large group of RNA viruses belonging to the family of Coronaviridae, and they display a high interspecies adaptability. SARS-CoV-2 is a large enveloped, single-stranded RNA virus with a positive sense, and it also entails 16 non-structural proteins, which are essential for viral replication, and 4 other structural proteins, namely the spike, membrane, envelope, and nucleocapsid, which mediate the cell invasion and immune response. Okay, so moving forward, I'll now talk about how the viral cycle in the cell occurs, talking about entry and receptor recognition and post-entry and exit. So the first thing that happens with the cell invasion is the viral entry, which goes receptor recognition, endocytosis, and viral membrane fusion. The post-entry steps include genomic RNA translation and replication, virion assembly, maturation, and exocytosis. So the first of those steps is the receptor recognition. The COVID-19 virus contains 2 ectodomains, a receptor-binding subunit S1, which ensures the viral binding to angiotensin-converting enzyme 2, ACE2, as referred to, receptor, and membrane fusion subunit 2. The S1-S2 domain has a furin-like cleavage site that is activated by furin, a cellular protease. This activation is responsible for the interactions between the S protein and the ACE2 receptor, as well as playing a role in the cellular fusion and intercellular spreading. ACE2 is a widespread protective enzyme, but it is also an entry point for the SARS-CoV-2 virus. The mRNA for ACE2 has a higher expression in respiratory tissues, but it is also expressed in epithelial cells of the salivary gland, intestinal, and renal epithelia or cardiac cells. Circulating ACE2 levels are higher in diabetic, cardiac, or obese patients, especially those with organ dysfunctions or treated with angiotensin-converting enzyme inhibitors, ACEIs, and the angiotensin II receptor blockers, ARBs or ARBs. This potentially explains why it is said that people with diabetes, heart diseases, or people battling obesity are at higher risk for COVID-19. Now, moving forward, I will talk more about the entry of the virus after the receptor recognition. So after the S1 subunit attaches to the cellular receptors, the virus enters the cell through a clathrin-mediated endocytosis, and it is then transported to early endosomes where the transmembrane protease serine 2, TMPRSS2, cleaves the SARS-CoV-2 S protein for a second time, which facilitates the cell entry and the fusion between the endosomal membrane and the viral S2 sequence. So, because of this means that the inhibition of TMPRSS2 could play a therapeutic role in inhibiting the virus's ability to enter the cell for replication. Because if TMPRSS2 were inhibited, then the cell or the virus would have a much harder time entering the cell, or it might not even be able to at all, which would definitely halt the spread of the virus from cell to cell. Okay, after the virus has entered the cell, the stages of the viral replication includes genome replication and subgenomic mRNA transcription, both which are mediated by the replication transcription complex, the RTC, a membrane-bound structure encoded by the virus. NSP12-RDRP also plays a large role in both of these processes, which I will talk a little bit more about later. But the final stages after entry of the viral cycle cover the viral translation of the structural proteins, the assembly of all the viral components into the endoplasmic reticulum, the recreation of the virions in the Golgi apparatus, and their elimination through exocytosis. Now, now that I have explained the whole cycle of the virus in the cell, I will talk a little bit about some possible treatments. As stated earlier, NSP12-RDRP plays a central role in genome replication and subgenomic mRNA transcription. This is because NSP12-RDRP is the SARS-CoV-2 RNA polymerase that mediates the replicase transcription complex, the RTC, as I stated. So without the NSP12-RDRP, the virus would not be transcribed and it would not replicate. So that's why there are multiple drugs that are targeting this factor as in the process to halt it. One of these drugs that targets NSP12-RDRP is favipiravir, and it is currently approved to be manufactured and sold in Japan. It is an RDRP inhibitor with a large antiviral activity factor. Inhibition of RDRP would diminish the activity of the RTC. This would largely reduce the severity of the virus because the RDRP is the key to the RNA viral replication, so it would inhibit the reproduction and spreading of the virus throughout the body from cell to cell.